Hi everyone, welcome to the Bioinformatics Coach channel. In today's video, I am going to show you how you can perform genome assembly on Oxford Nanopore data. So what we are going to do is to first download the example data, and then we are going to trim that data using the tool called Porechop. After trimming, you will then perform the genome assembly on the trim data using the tool Flye. So this is the GitHub page for Porechop, the tool we'll use for the trimming. So you can just go and just read about it here. I will leave the link in the description box. This is the GitHub page for Flye. So you can also just go and just read about Flye here. So the link to this page will also be given in the description box. So just use those links. So these are the tools we'll use. Now, in terms of PC requirements, you need to have a Unix-like environment. That is what I used anyway. So Linux or macOS will be fine. I've not really used Flye on a Windows machine, so I'll not be able to tell if it works or not. Probably you may have to contact the developer, but it's going to work on a Unix-like environment. So just make sure you have one: Linux, macOS, etc. So that's it. Aside this, you should also have Anaconda installed on your system. Okay, so I have a separate video that shows you how to install Anaconda in a Linux environment, so you can just watch that. The link is in the description box, so please do that. So that is it. Please note that Genome Assembly is a computationally intensive and expensive process, so you need to have a high-end PC, a PC with a good size of RAM to help do this. So the best way, or the advisable way, is to just use a Linux server that has high computational power to do some of these tasks. So just make sure you have a fairly good PC you can use for this activity. That's what I want to say. So those are the requirements. Now, back to our data. So the data we are going to use, I got it from this paper here. I will leave the link to this paper in the description box as well. So with this paper, they perform whole genome sequence on Mycobacterium monstrans isolates, and they had Illumina sequences and also Nanopore reads. So those are the two platforms they use. But our focus today is on the Nanopore reads. So that is what we'll use. The data was dumped in the SRA database, so you can just go and just download it. This is our session number for the Nanopore reads. So I'll just copy it. Also, you should also copy as well. We only need this as session number here. So just copy it and then go to your browser and then just visit SRA. Okay, so it is the database. So I will leave the link in the description box. So just visit it and then just paste your session ID here and then just search. Let me chip in that there is also a command line tool to help you download data from SRA database. So if, let's say, you have multiple files or you are dealing with several accession IDs, you can just use those tools to help you quickly download instead of using the browser. But because we are dealing with just a single data, we are just going to use the browser to do it. So paste here and then just search. And then after searching, you'll be sent to another page. Here, just scroll down here to the button here, and then you see this table here. You use this accession number here, this ID here, the link here. So just click on it, and then you'll be sent to another page. So our focus is to download the data. So we'll just go to the Data Access tab here. So just click on it, and then you'll be given the download links. So that is what we are going to use. So this one worked for me. This one here, the one I've highlighted, worked for me. So that is what I will use to get my data. So I'm just going to copy the link here, and then I'll go to my terminal and then download. You can also click the link anyway and then just download using your browser. But if you are using a remote server where you don't have access to browsers, then you just have to use the link and then just download on the terminal. So I will do mine on the terminal. So back to our work. So I'm on my terminal now. So what I want to do is to create a directory that I will perform my actions in. It's advisable to do that. It helps you to organize your files, your activities, and other data that you'll be generating, and also helps you to separate them from other things you've already done on your PC. So I'll first create a directory called Genome Assembly, and then I'll cd to it, and then I'll just do an ls just to check to see that there's something there, but it's an empty directory we created, so there'll be nothing there. So from here, I'll just do a wget to just download the file. So I'll supply it with the link here, and then I'll download it. Aside wget, you can also use curl and or other utilities to help you download stuff by supplying them with a link. So just look at one which works for you. But wget is popular and it's available on almost all Linux platforms. And you can also install on macOS, so you can just use that to do the download. So just download and then let's get some stuff done. So let's do it. So download has been done, and so you can see it here. So do an ls, and you see that the file is already there. So let's clear the screen, and then let's proceed. The next thing to do is to install the tools using Anaconda. First of all, if you have installed Anaconda, we need to add channels. So you add Bioconda and then Conda Forge channels. So you say, now we use this one here. So we say conda config add channels. Let's add Bioconda first. Let's add a second one, which is Conda Forge. Let's confirm that the channels have been added. So we say conda config show channels. So this will list the channels. So we have the two here. The default one comes packaged with Anaconda, so we just leave it as it is. So we are done. The next thing to do is to create an environment and install the tools that we are going to use in them. So we say conda create -n. So this will create an environment for us. So we say Genome Assembly, and then we specify the tools. So we have Porechop, and then we also have Flye. So these are the two tools that we are going to use to do the installation. So just use this and then just get the stuff done. So the -n indicates the name of the environment, and we have our tools here as well. So we just do all of them and install. So let's execute this command and then get the installation done. So let's confirm by typing Y. Okay, so installation has been done. So let's clear the screen first. Yeah, so it's done. So if the installations are done and we want to use the tools, we need to activate the environment. And so that is the next thing we are going to do, to activate the environment. But then if, let's say, you have forgotten the name of the environment, you can still list and then look at the one which we are going to use. So let's say conda env list. This will list the environments. So notice the one we created, Genome Assembly. So that is what we are going to use. So we will say conda activate Genome Assembly. Please note that this procedure will not be necessary if you have already used a different way of installing the tools. So once you have the tools available, that is fine. Okay, so now let's just test. Let's start with Porechop. We are just testing Porechop. So it's there. So these are the options how to use it. Let's try Flye. It's also there. So the tools are already there and they are ready to be used. Let's clear the screen. So let's go back to the GitHub pages. Let's start with Porechop, it has requirements. I think that one I already said that. Yeah, so even this one says Linus or Macro, so those are there. And then just go down, you see a number of examples as to how Porechop is used. So this is a basic adapter training, and that's what we are going to do with this tutorial. We'll just use a basic one. Now Flye too has usage. So if you go to Flye, let's go to Flye, yeah. You just look at the documents and usage and you see how it is used. So just the options here. So just go and read about all these things there. So now back to what we want to do. We are going to first trim. Okay, so let's do an ls to get our data. This is our data. Okay, so we are going to trim it. You can also decide to rename it just to make it fine, but for now let's just stick to these ones here. So let's just give it a name. Let's say, so let's just rename it. Let's say move all menu to, let's say, Nanopore data.fastkill. It's a fastkill file, so we are just naming it this way, just for consistency sake. So we've renamed it. Let's do an ls, so it's there. So now we are going to do the trimming. So we are using Porechop. Before I proceed, let me also say that I use the words faster and fast A interchangeably. These all refer to the same file format. So what we will do is to say Porechop -i, which will be the input file. And because we renamed it, we will say Nanopore data.fastkill. And then we specify the name, the output name. So we use -o. So let's say Nanopore data.trimmed.fa. We are going to trim and then save it as a faster file or a fast A file. Let me put it that way. So fast A file, and then we just indicate with formats as fast A. Okay, so the format here will allow Porechop to use the appropriate algorithms to convert or transform the data. Convert, that will be the appropriate way to convert the data after it has trimmed this to faster file. So this is how it is. Now let's add threads. So let's say 10. So threads, multi-thread, and so this allows the work to be split into smaller chunks and then assigned to different CPUs on your computer to do the work. So it makes it faster in some way. So I'm using 10, but I can go more. I have more computational power, but let's use 10 for the meantime and then just trim it. So we do the trimming now. So the trimming has been done. So let's clear the screen first. Let's do an ls and you will see the data here. So notice I specify as the outputs nanopore data.trim.fa. I usually try to also include some details into files that I generate using these tools, just to help you to keep track of what is happening. So you can just use that or adapt a naming convention as well. So this is .trim, so I know that it's a trimmed file. Okay, so it's a faster file. Let's do a head. Let's just do head. Nanopore data.trimmed, just to look at some of the files. So this is a faster fast A file. So let's just, so you can just explore, just look at how it is, but these are things that we do later on. So let's proceed. So let's clear the screen again, do an ls, and we have it there. So the next thing to do now is to perform the genome assembly using Flye. Okay, so you just do the assembly. So you include some parameters and I will explain them as we go. So let's call Flye and then we indicate nano raw. So this tells Flye that the data we are inputting is a Nanopore data, but it's a raw data, raw reads. And then we specify the file. So this was the file name. So we say nanopore data.string.fa. And please take note that Flye can also assemble reads that are in the FASTQ format. So either way it's fine, whether FASTA or FASTQ, you can just use any of those formats to do it. So now we've specified it. Now let's look at -o. So -o is output directory. So this is the name of the directory that Flye should create and then place your contigs in there. So let's say -o, let's say let's just say assembly. I think that's fine. Assembly. Let's put it that way. So this directory will be created for us. You can also use any name of your choice. Here is directory that we use. Then let's add some additional options. One of the options we are going to add is the genome size, which we say -gen, and we say 5.6 M. Because it's a Mycobacterium ostrans isolate, the genome size is around this, so we just put it this way. You can just experiment with the numbers, but this should be fine with the organism we are dealing with. And then we also indicate -t for threads. Okay, so threads, let's just say 10. And then the other option we are going to add is polishing. So we want to include polishing. By default, Flye does one iteration polishing. So we are going to increase it. I think the maximum, the last time I checked, the maximum was 10 or so, but let's use let's make it 2. So we say -2. I mean, we are making it 2 so that you can quickly get your assembly done, so that you can just do other stuff. But later on, you can just experiment with the numbers and see how it improves your assembly context. So this is how it is. So once we are done with this, we can then run it. So yeah, so let me also stretch on this. So here, because the genome size is MB, that is why we use M. If it's a different unit, you just have to make sure you put it there. So it's M, and then yeah, so that's what I would say. But just try, if there are other organisms which has higher genome size, just make sure you put the appropriate units and appropriate values there as well. So just take notes. And let me also say that with Flye, you can also assemble PacBio reads, and that is why this here is important, because this tells Flye which sequencing platform, and it will use the appropriate algorithms to do the assembly. So it's important to specify which sequencing technology you are using here. So that is what we have to do. So now let's just run this tool. So you will see that the assembly process has been started. So just wait whilst it runs. Okay, so genome assembly has been done. It's complete. And that is why you have this message here. Okay, just to let you know that Flye has finished everything. Now, once Flye completes the assembly, it also gives you some statistics here that you can use for QC purposes. So N50 fragments, total length, and mean cover order. These are things that we will look at in detail, but it helps you to really confirm that indeed the organisms that you are dealing with are really what you think they are by looking at some of these statistics. And it will also help you to detect maybe there were some anomalies, or maybe you could also improve the genome assembly. So these are things that you just have to look at, but I'll try and also look at topics around that area of QC on genome assembly data. So this is how it is. So it will also tell you where it has saved the assembly contigs. So the final assembly, this is the file. Okay, so this file will contain contigs. So the contigs are short sequences that you can later process to get your scaffold. Okay, so let's go into that. Let's clear the screen first, Oh, and before I go, let me also say that notice that we had what? Polishing. We did 2. We did I equals 2. And so we had 2 iterations, which is this one. Notice this one here. Polishing genome 1/2. Polishing genome 2/2. So, and that's how it is. So you can increase it, and the higher the number, the longer it will take for the assembly to complete, because these are iterations. So that means it will have to go over and then redo some of the stuffs again. So let's clear the screen. Let's do an ls, and notice the directory we created. We didn't create it, by the way, so we specified when we were calling Flye to do the assembly. So this is where our interest is going to be now. So let's ls that directory. You have a number of stuff there. Okay, so let's look at the log here. So you can just do cat assembly/flye.log. Yeah, so this is the same thing as what we saw. So these are logs you can just go into and investigate. So some of these things are there so that you can investigate and see what really happened in the process of assembly. It helps also for debugging purposes and for other stuff. So that's how it is. Yeah, assembly statistics. Yeah, so you have it here. But there's also a tool that can help you to do a detailed statistics. It's called Quast. So if you want to look at your assembly quality and then some statistics, you can just look at that tool. I will leave the link to that tool also in the description box. So use that to get some QC done for your assembly. So that's how it is. So let's do ls again to assembly. So there's a file you are interested in. There's a context. So let's say head assembly. There's a directory, and there's the file .faster. Okay, so there's config 1. You can also cd into it directly and then just do the head, but it's fine, just experiment. So how many contigs do we have? This is a FASTA file. Okay, so for FASTA files, what happens is each sequence begins with this sign here, the greater than sign here. So if you want to know how many contigs are present or how many sequences are present in a FASTA file in general, you can just count the number of this and you get it. So let's say you can say grep and then assembly.fasta and then just do wc-l. This will count how many contigs. So you have 66 contigs here for us. So that's how it is, but it's fine. So the purpose was just to get the genome assembly done, and now we have what we need here. So, and we also did some polishing, so it has been very good. So I believe this has been helpful, and then you can now proceed to do your other stuffs as well. So after genome assembly, what's next? There are lots of activities and analysis you can do after performing genome assembly. And here I display a non-exhaustive list of some of these activities. I have also covered some of these activities in detail in separate videos. And if you want to watch those videos, you can click on the playlist that I'm showing right now. Thanks for watching this video, and I'll see you in the next session. Goodbye.